Hey guys, welcome back to the channel. I'm going to be starting a new series where in under 10 minutes I will be going through each and every IB biology topic. So starting things off with 1.1, which is an introduction to cells. So we go over cell theory, which states that all living things are made up of cells. This means like bacteria, prokaryotes, all of that. They're all made of this fundamental component called a cell. And with the exception of viruses, they're a bit different. We'll go into that later. Cell is the smallest living unit of life. Cell is the smallest unit of life is the second point of cell theory, and this concerns the fact that anything smaller than a cell, like an organelle of a cell, is technically not living because it won't be carrying out the functions of life, which we'll get into soon. Cells only arise from pre-existing cells. This is talking about spontaneous generation not being a thing. You can't make cells out of nothing. Cells always come from pre-existing cells that are already there, like through divisions of mitosis essentially, and or meiosis. And there are exceptions to cell theory, and there's first one is striated muscle fibers, which are very long, so they can be like I'm pretty sure they can be about 30 centimeters long, so like a standard ruler, and that's quite large for a cell. So it's an exemption. Also, it has lots of nuclei, so it's against the notion that a cell is a single component of life. And similar to this is the aseptate fungal hyphae, which has these filamentous hyphae regions which separate the hyphae cells. So instead of acting like one, it's actually got partitions, and the single cell is sort of like a colony of cells. So it's an exception to the fact that it's the smallest unit of life, and it's suggested that these smaller components are even smaller parts. Acetobularia is an exception because of its large size. It can go up to like 7 centimeters. So that's going against the notion that cells are quite small. And when we talk about cells and their ability to be considered living, we go over the functions of life, and the way I learned it was Mr. Sheng. So metabolism, reproduction, sensitivity, homeostasis, excretion, nutrition, growth. So metabolism refers to the going over the metabolic reactions of living things, so such as cellular respiration. Reproduction means that you can make more of yourself, so cells can go reproduction through sexual and asexual pathways. Asexual, such as binary fission, sexual, such as meiosis. Sensitivity means that you can respond to the environment outside of you. Some cells, unicellular organisms, have light receptors on their cell membranes, which allow them to move towards light. Homeostasis, maintenance of an internal environment. So you got osmotic pressure, you got salt concentration, whatnot. You can maintain that through various mechanisms such as contractile vacuoles. Excretion means you can remove waste from the cell, so this could be through carbon dioxide diffusing through membranes. Nutrition also relates to excretion. It's about getting good stuff in essentially. So you got the glucose and all that, glucose, nutrients, and also oxygen through membranes, so you can get through the endocytosis. Growth means that a cell is able to assimilate mass as it conducts, as it takes in nutrition. So this could be cells growing larger. And the main cells we need to know about are the paramecium and cendesmus, which you might have to label with the functions of life. So paramecium is the heterotroph, and cendesmus is the autotroph. Heterotroph as in they are like the animal cell, and this one's sort of like the plant cell. Next point we concern is the surface area to volume ratio, which is got a bit of maths to it, so it's just the surface area and volume represented as a ratio. Surface area is important because it represents the ability of a cell to exchange material with its outside environment. So the larger the surface area, the greater the exchange. Volume is important because it is representative of um, how much resource consumption and waste production occurs in a cell. So the larger the volume, the more nutrients it will need. So when surface area to volume ratio is high, that means lots of exchange can occur for a relatively small amount of waste production, resource consumption. So that's good. But surface area to volume ratio is low would mean that there's not enough exchange happening for the heavy consumption and waste production in a cell. So that would be bad. So surface area to volume should be high. And what you should know is that when cells grow, so looking back at the functions of life, surface area to volume ratio falls because the surface area increases at a lower rate than volume. So that's kind of bad. So we need to go over that. That's why cells are also very small. They don't grow bigger, because if they did, the surface area volume ratio would be too small and cells would die. Next up, we have magnification. And the key here is to remember this formula. I call it IM. This is what I learned at school. And this is a triangle like this. So I M. And what this means is, to work out the image size, you multiply the actual size and the magnification. And to get the magnification, you get the image size divided by the actual size. Actual size, you divide the so you get the image size and divide by the magnification. So that neat triangle helps you do all that in an exam. Next up, we have emergent properties, which are properties that arise from cells working together as one. So cells are bundled together to form tissues. Tissues form organs. Organs form organ systems, and organ systems form organisms. So when tissues are specialized and work together, they can conduct a function that they couldn't have conducted by itself. And this is one of the advantages of being a multicellular organism as opposed to unicellular, because of course unicellular organisms cannot do the emergent properties by itself because it's single cell. And relating to this is the notion of specialization, which refers to the expression of some genes but not others. So genes are in the form of DNA, and some of the genes are going to be transcribed, which we'll get into later in topic 7 and or topic 2. We get that's when DNA is converted to protein, or DNA is read out and translated to protein and transcribed. So the formation of proteins through transcription and translation is going to be only available in a certain type of chromatin. This is called euchromatin. So euchromatin is the kind that is not very condensed. So you can imagine the cells have this DNA, and then some of it's condensed, some of this could be very lightly there, and then the super condensed one would be hard to access, so those genes would be repressed, and the lightly condensed one would be easier to reach, so those genes would be expressed. So that's how differentiation works, and you get desired characteristics that are suited for cells' functions. And the last part about this topic is the notion of stem cells, which have two characteristics. They can divide and replicate infinitely, and they can differentiate into any type of cell that it desires. So these are especially useful in replacing cells of cells that are malfunctioned. So 3 examples that you'd have to know about are Stargardt's disease, which is in the eye. So what happens is some rod cells in the receptors do not function properly. So you'd culture stem cells and extract them and insert the stem cells in here into the retina, and vision usually improves. Next up, we need to know about Stargardt's Parkinson's disease, which you might have heard of. It's quite popular disease, but what it is concerned with is nerves. So I've tried to draw a neuron here, and what happens is the nerves are dead. And the fun fact about nerves, well, it's not fun, but is that they cannot self-replicate. So when a nerve dies, it's dead forever. So stem cells are really important in reviving these dead nerves. That'll help allow a person with Parkinson's to have better control over their movements and not have rigid movements. Another one you need to know about is generally to do with cancers, and the one I remember is leukemia. So leukemia involves bone marrow being extracted from the spine generally, and that bone marrow contains stem cells. And what happens is the cancerous cells due to leukemia. So leukemia and cancer is due to uncontrolled division of the cell. So lots of cells forming tumors and all that, so rapid division that we need to get rid of. So chemotherapy is done to get rid of those bad cells, and the stem cells that we took out, so the stem cells that we took out from the bone marrow will be injected back into the body, and they'll be able to function normally. So that's pretty much the stem cells uses. And you need to know about the ethics of using stem cells, so whether it's from an embryo or an adult, or it could be from a umbilical cord. So you need to know about which ones have certain advantages. So some could divide more, so especially the earlier stages. So the umbilical cord and embryo can divide a lot more, but then embryo gives up the notion that whether you're killing life. So you got to consider these factors and the ethics. You got to talk about religious stuff and whether rich or poor get the embryos. And essentially that's topic 1.1. Thanks for listening, and this is done under 10 minutes. Next I will be doing 1.2.